Here we go with the last neurology lecture. So these are going to be our special senses. We're going to go with the eye and vision, then taste and smell, and then hearing and balance. So first off, your eye and vision is going to encompass 70% of all sensory receptors. So 70% of all sensory receptors are going to be present in the eye. About half of your cerebral cortex is going to be involved in processing that visual information, and the eye is going to be protected by a cushion of fat in the bony orbit. So around your eyeball there's a little layer of fat kind of protecting it, so if you take a blow to the eye it's not just pushing up against the bone. With the eye, we're going to start off with the accessory structures, so the eyebrows, the eyelids, the conjunctiva, lacrimal apparatus, and the extrinsic eye muscles. So first off, the eyelids, they're going to be separated by the palpebral fissure, which is going to be just a split between your eyes, so it's just this guy right underneath the eyelashes. There's going to be lacrimal caruncles. These are going to be elevations in the medial commissure. Let me show you this. Oops, wrong way. Here's your lacrimal caruncle. It's a little bump in the midline towards your eye. Next one, you're going to have your levator palpebrae superioris. So here it is, levator palpebrae superioris. That's going to be to lift the eye lid. You're going to have your eyelashes. Those will have nerve endings, which will help you with blinking to help protect the eye. And you're also going to have lubricating glands. Those will be your tarsal glands. Those will be associated with your hair follicles. And then you'll have your eyebrows. These will overlie the supraorbital margins, and these help with shading the eye. So here's your palpebral fissure. Here's that lacrimal caruncle. Here's your scleral conjunctiva. And you're also going to get bulbar conjunctiva. So when you hear of conjunctivitis, it is irritation of this conjunctiva. And you have scleral, which is the white, and it goes all the way down into the bulbar conjunctiva, into the conjunctival sac down here. So what can happen is when you get conjunctivitis, it's inflammation of that conjunctiva. It turns red and it's swollen and itchy and stuff like that. Okay, with the lacrimal apparatus, you'll have lacrimal ducts. They're going to connect to the nasal cavity. They're going to make a dilute saline solution. It'll be mostly water, but it'll be mucus, antibodies, and lysozyme. They're all there to protect your eye from infection. Your eyes are constantly open, and it's a gateway for microorganisms to enter the body. As you blink, it is going to spread the tears towards midline, and the tears are going to get drained via your lacrimal canaliculi and puncta. So here we go. Here's your lacrimal gland. It's going to create tears. They're going to go through the lacrimal ducts. They go through the eyes. You blink, they move towards midline. Alright, it's going to get drained by the lacrimal canaliculus. Okay, it goes through the nasolacrimal duct. And you ever notice, like when you're crying, you feel like your nose is running? Here's the reason why. Is that as the tears are draining, they actually drain through your nose. And then your conjunctiva. So it's going to be a transparent membrane. You'll have palpebral conjunctiva. That's going to be on the eyelids, and the bulbar conjunctiva is going to actually be like the sclera, the white of your eyes. This will produce lubricating mucus also. With your external eye muscles, there's going to be 6 of them. You'll have a medial rectus, which will go midline. As it contracts, it pulls the eye medially. You'll have a lateral rectus, which as it contracts, pulls the eye laterally. You'll have the superior oblique, which will depress the eye because it goes up here. It's going to pull in this direction. It's going to depress the eye and turn it laterally. You'll have an inferior oblique, which will elevate the eye and turn it laterally. And you'll have a superior and inferior rectus. Superior rectus pulls it up, inferior rectus pulls it down. Your medial rectus, superior rectus, inferior oblique, and are all going to be controlled by your oculomotor nerve, cranial nerve 3. The lateral rectus is done by your abducens nerve, and the superior oblique is done by your trochlear nerve. Okay, with the eyeball, you're going to have a couple layers to it. There's going to be a fibrous outer coat, a vascular layer, and a sensory layer. It'll have an internal fluid-filled cavity that will be broken up into anterior and posterior segments. The anterior segment is filled up with a aqueous humor, which is a watery-like substance. The posterior segment is filled up with vitreous humor, which is like jello almost. So the outer fibrous layer, the outermost fibrous layer, is going to have two parts. The sclera, which is the white of your eye, and the cornea, which is going to be the transparent portion of your eye where light's going to pass through. The middle layer, the vascular layer, is going to have three regions to it. It's going to have the choroid coat, the ciliary body, and the iris. Choroid coat is going to be the posterior portion of the uvea, and it's going to supply blood to all the other layers of the eyeball. It's going to be brown in color, and it's going to help absorb light to prevent confusion. So if you took human bio, when you dissected the eye, the layer that was like kind of crazy colored and brown or black looking was going to be this, and it's there to almost make the eye like a black box and to absorb any extra light. So you're not getting all kinds of crazy extra input into your eye. Ciliary body is going to be a ring of tissue around the lens. It's going to be smooth muscles, which are our ciliary muscles, and they're going to have it's going to have this thing called the ciliary zonule, which is the suspensory ligament. It is going to be the attachment from the ciliary muscles to the lens itself. So those muscles can constrict and relax to change the shape of your lens, to change the way the light is coming into your eyes. And then last is the iris. So your iris is going to have a central pupil, and that is going to be able to close and open. So when it closes, you get your sphincter papillae, which is the circular muscles. That'll close it. Or if you're going to open it, you use your dilator papillae, which will open the iris. For close vision, you use the sphincter papillae because you don't need as much light getting into the eye. For further vision, you need more light, you need to process more information, so you use the dilator papillae, and that'll open up the eye. The iris is also going to be the colored portion of the eye. Okay, so here's the iris, here's the pupil. It can close or it can open up. Here's your lens. Here's those suspensory ligaments in the ciliary body. So if this muscle can change its shape, and it can flatten out the lens, or it can contract and the lens can become more round. Here's your sphincter pupillae muscle. If that contracts, it closes up the eye, the iris. Here's your dilator pupillae muscle. If that contracts, it will open up the iris. In fight or flight, the dilator pupillae contracts. Your pupils open up big and wide. Why? You want to take in as much information about everything around you as possible. Parasympathetic, rest and digest. That sphincter pupillae will contract, and you don't need to be taking in as much information. And then next one is our sensory layer. Within the sensory layer, there's going to be two membranes, a pigmented layer and a neural layer. The pigmented layer is the outer layer. It's going to be there to absorb light, and it's going to be kind of a darker, shiny color, and it will also store vitamin A. With the neural layer, that is going to be where your photoreceptors are, and this is where the input of information actually begins. So looking back here, you're going to have your retina, which is going to be the neural tissue that will take in information. On the retina, you have this thing called a macula lutea and a fovea centralis. Fovea centralis is going to be the sharpest point of vision for you. Other thing you're going to have is this right here, your optic disc. This is your blind spot. This is where blood vessels and the nerve are actually entering the eye, and there's a little hole where there's no neural tissue, so with no receptors, so you're not going to have any vision at that small spot. With your vision, you have two types of receptors. They are photoreceptors. You have rods, which will see black and white, and you have cones, which see color. Rods are going to be more numerous, and they're going to be on the outer part of the eye, away from the macula lutea. The cones are going to be found near the macula lutea and the fovea centralis. So if you remember, I said this fovea centralis is going to be where your vision is sharpest. So we're going to want to take all the information and bring it, light passes through the lens, and try to center it right on that fovea centralis. In dark light, when you see like shadowy type images, that is, you are using your rods, so those will be seeing black and white. In bright light, you will be using your cones. If you are in like a dark room and you flick on the light and your eyes hurt for a moment, the reason why your eyes hurt is that what needs to happen is you need to burn out the rods, basically. Those rods need to get overstimulated and just shut down, not completely shut down, but basically shut down, so you can use your cones and function in the brighter light. Okay, with the internal chambers of the eyes, we were talking about these before. It's going to be broken up into a posterior and anterior segment. The posterior segment lies behind the lens. The anterior segment lies in front of the lens. Okay, the anterior segment will be broken up into anterior and posterior chambers. The posterior segment is going to be there to support the posterior surface of the lens, and it's going to transmit light, and it is going to hold the retina firmly against the pigmented layer. If that is not happening, you start getting issues with your vision. With the anterior segment, that is going to be broken up into two chambers. There will be your anterior chamber, which is anterior to the iris, and there will be the posterior chamber, which is going to be posterior to the iris. So this part right here is the posterior chamber. This is the anterior chamber. This is going to be filled up with aqueous humor, watery fluid. Humor means fluid. And this is going to be filled up with vitreous humor, and vitreous humor is like jelly or jello. Okay, your anterior segment is going to contain the aqueous humor, like we just said. It's going to be a plasma-like fluid continuously filtered from the capillaries of the ciliary process. It will drain via the scleral venous sinus, which is the canal schlem, and that will be found in the anterior chamber of the anterior segment. Let me show you. So here's where we're going to be making the aqueous humor. It travels up and over the iris, and it's going to drain anteriorly through the canal schlem. Okay, so aqueous humor is formed by filtration from the capillaries of the ciliary process. It flows from the posterior chamber through the pupil, and then it drains out through the canal schlem. We have an issue called glaucoma. You guys have heard of glaucoma before. Basically what's happening with glaucoma is your canal schlem is not properly draining your aqueous humor. So what will happen is the pressure builds, it pushes back on this lens here, and this is in a constant state, so we push back on that vitreous humor, and that pushes on your blood vessels and on your nerve. So if you're pushing on those, all of a sudden they're not getting all the nutrients they need, and they're getting messed with, so it can drastically affect your vision. With the lens, it's going to be a biconcave disc. It will be transparent, flexible, elastic, and avascular. It'll allow precise focusing of light on the retina. The cells of the epithelium can differentiate into lens fibers that form most of the lens. Lens fibers are going to be filled with a transparent protein called crystalline, so that your lens is basically like a crystal, but it still has these transparent, flexible, and elastic properties to it. If your lens as you age, what can happen is you could get clouding of the lens, and that the most common things that's going to be associated with is going to be diabetes mellitus, heavy smoking, or frequent exposure to bright light. Okay, with the refraction of light passing through the lens, the gist of what you need to know is that the image passing through the lens is going to be inverted, which means it's going to be upside down and reversed. When you see something and it is upright and what you're actually seeing, the reason why it is that way is that your brain actually processes the information and flips it up so you perceive the image as being upright, proper. So check this out. So whatever you're looking at is upside down and reversed. So whatever was on top will now be on bottom. Whatever was in front will now be in back. That is the information that travels into your eye because the lens flips it all around. The reason why you see it as normal, like I said, is because your brain processes it and tells you this is normal. So see, we have our orange box and our light blue box. Here the light blue box is on the bottom and in front rather than on the top and in back. So it's upside down and reversed. What was on top is in bottom, and what was in back is now in front. So when we focus the light on the retina, the pathway of light entering the eye, I want you to know this and remember this. It is going to go through the cornea, the aqueous humor, the lens, the vitreous humor, the neural layer of the retina, and that will be with our photoreceptors. Light is going to be refracted at the cornea and when it is entering and leaving the lens. But we can change the shape and curvature of our lens to allow for fine focusing. That's the way you can look at something close and far away. So, if light rays from a distant object are going to be nearly parallel at the eye and need little refraction beyond what occurs in the eye at rest. So that means that our ciliary muscles aren't going to need to be contracted and they're going to let the lens stay flattened. When you are seeing stuff past 20 feet away, that is going to be the situation. So you'll call that emtropic. It's going to be a normal eye. The ciliary muscles are relaxed and you're going to be pulling on these suspensory ligaments and flattening the lens. So these guys will be coming in basically parallel. When we're looking at something close, accommodation is going to occur. So you're going to change the shape of the lens. Look at, so it's not coming in here. Here it was coming in nice and parallel. It's basically coming straight in. When you look at this picture, it goes up and down. So it's going to need to be refracted. So accommodation occurs. This is your near vision. If you lose your near vision, we call it presbyopia. What happens is you're going to have constriction of your ciliary muscles, which will loosen the suspensory ligaments, and that'll allow the lens to bulge. So some problems that you can have with refraction or the lens of your eye are myopia, which would be nearsightedness, hyperopia, you're farsighted, or astigmatism. With astigmatism, you're going to have unequal curvatures in the lens or in the cornea. So you're not going to see, everything will stuff can seem like it's slightly blurry to you, whether if it's close or far away. With our photoreceptors, like we said, we have rods and cones. Rods are for black and white. Cones are for color. The outer segment of each one is going to contain visual pigments. It's going to be a molecule that will change shape as it absorbs light. Okay. Within the retina, there'll be light-absorbing molecules. They'll combine with 1 of 4 proteins, or opsins. Okay, and those will form our visual pigments, and we're also going to be helping out with synthesis of vitamin A. So check this out. We talked about this in the last lecture. Right side of your brain will be taking care of vision to the lateral portion of the right eye and the medial portion of the left eye. I'll say it again. Right side of the brain is going to be taking care of sensory input from the lateral portion of the, sorry, I should be doing the right eye if I'm going to be talking about it. So with your right side of the brain is going to be taking in sensory information from the lateral side of the right eye in the medial side of the left eye. Left side of the brain is going to be doing the lateral portion of the left eye and the medial portion of the right eye. Why is this important? This is important because your visual fields overlap. This is going to help with the yolking of your eyes, and it's going to cause your visual fields to overlap. These nerves will pass through the optic chiasm. So that's this guy right here. It's going to connect your right and left eyes together. So when you close your eye, if you close like I'm closing my right eye right now, I'm only losing, it tops maybe 25% of my vision. It's off to the right side, but I'm not losing a terribly huge amount of vision. Close my left eye, like I said, I'm losing like 25%, but it's just going to be a little bit, not everything. With depth perception, in order to have proper depth perception, if you have vision in both of your eyes, you need to be using both your eyes to have proper depth perception. If you lose vision in one of your eyes, over time your body can get back the depth perception, but it really helps to have the two eyes so you can get a better three-dimensional vision. Now we're going to go into taste and smell. Taste and smell are going to occur through chemoreceptors that are going to respond to chemicals in aqueous solution. First here we have we're going to have sense of smell. The organ of smell is going to be the olfactory epithelium in the roof of the nasal cavity. So you'll have your olfactory tract with the bulb with those olfactory hairs coming down. You'll have bundles of axons or olfactory receptors that'll form filaments of the olfactory nerve. So basically, air comes into the nose, comes up, and it interacts with these. You're going to have chemical reactions going on. So you're going to have dissolved odorants binding to receptor proteins in the olfactory cilium. So this is just going to be dissolved chemicals. It's just going to be chemicals floating in the air that come in contact with these. Each one will respond to several different odors, and each odor will bind to several different types of receptors. Those receptors activate, they send information to your brain, and you perceive it as whatever it might be. A flower, a dead animal, I don't know, a yummy steak that's cooking. You'll process that to whatever your brain has decided to perceive the information as. How does it decide to perceive the information? By which receptors are being stimulated and which type of receptors are being stimulated. With our sense of taste, you're going to have taste receptors, or your taste buds, right? Your taste buds are going to be found on the tops of your fungiform papillae. If you look at it microscopically, I see these if you see a better picture. We'll go more in depth, not too much more in depth, but we'll talk a little bit more about taste in the digestive chapter. But we'll find them on the tops of our fungiform papillae, and also you'll find them in the sidewalls of our circumvallate papillae and the folate papillae. So here's your circumvallate papillae. It's described as being flask-shaped. Okay, there will be basal cells, and there's going to be gustatory cells. The gustatory cells are going to be the actual taste receptors. So these will be these guys right here, the taste buds. So you salivate, saliva is made in this little circumvallate, a valley, and it will interact. It'll mix up with the food. It'll come back in here, and those chemicals will interact with these taste buds, and you'll perceive it as taste. So with your taste, you have 5 types: sweet, sour, salty, bitter, and umami. Different parts of your tongue will have different parts of taste. So this is a nice little picture. Just shows how here's the sweet and salty section, here's the sour, here's the bitter. Okay, umami is going to be savory. With your sweet taste, you're going to have chemoreceptors that will be sensing sugars, saccharins, alcohols, some amino acids. With the sour taste, you're going to be having chemoreceptors that are going to be responding to hydrogen ions. Or salt, they'll be responding to metallic ions or metal ions. Bitter will be alkaloids. And with the umami, it's going to be amino acids, glutamate, and aspartate. So with taste transduction, basically, the anterior two-thirds of the tongue is going to be covered by the facial nerve. The posterior third is your glossopharyngeal nerve, and the wall of the pharynx is going to get some from the vagus nerve. With your sense of taste, 80% of your sense of taste is from smell. So think of smokers. They sit there, they're smoking a cigarette. It's very, very hot. There's tar and all kinds of bad chemicals coming out of the smoke. As they're breathing, that smoke comes into their nose, and it is just burning and frying and killing off these nerve cells here. These nerve cells are resilient, so some of them will continue to survive, but over time a lot of them die. Why do I describe that? Because think of smokers. Smokers typically are going to like very, very spicy or bland foods. Why? Because they don't have as good of a sense of smell as other people, because they've killed off those nerves, and they're going to need something to stimulate the nerves, or they're not going to like much taste at all. It's also going to be a combination of thermoreceptors, mechanoreceptors, and nociceptors in the mouth to help influence our taste. So some people will decide if they like the taste of something based on its texture, while others will decide if they like something just strictly based off of their taste. Okay, temperature and texture will enhance or detract from the taste of food. Now with hearing, we're going to go hearing and balance. So with that, we're going to have our vestibule and our cochlea, or our semicircular canals and our cochlea. Semicircular canals are mainly for balance. Cochlea is going to be for hearing. So starting to the ear, there's going to be three parts. There's going to be an outer ear, a middle ear, and an inner ear. The external ear is going to be involved with just hearing. The internal ear is going to be involved with hearing and balance. Equilibrium is going to be your balance. Receptors for hearing and balance are going to respond to separate stimuli, and they're going to be activated independently. What do I mean by that? You could be standing in a silent dark room and spin around in a circle and not hear a single thing, but you could be activating your balance or your vestibular receptors and not activating your hearing. Or you could be sitting perfectly still and not using your balance receptors but using your hearing. So with the external ear, we're going to have the helix, which is going to be the rim. You'll have the lobule, which is going to be the bottom, which is going to be your earlobe. You'll have your external acoustic meatus, which is going to be a short canal that is going to be lined with hair and skin and ceruminous glands. Ceruminous glands are going to be modified sweat glands. They make cerumen, earwax. And those will lead up to the tympanic membrane. Once you pass through the tympanic membrane, you get into the middle ear. So the tympanic membrane, like I said, is the division between the external ear and the middle ear. It's going to be a layer of connective tissue that'll vibrate based on sound. And as it vibrates this little guy right here, which is going to be one of your auditory ossicles, and we'll talk about them. That's going to be the malleus. The middle ear. So here we blew up. Whoops. Here we blew up that picture of the middle ear. So sound waves come in, they vibrate the tympanic membrane. Alright, so the middle ear is a small air-filled mucus-lined cavity in the temporal bone. It does not typically have fluid inside of it. If it has fluid inside of it, there's usually something going on. Laterally will be the eardrum. Medially, we will have a round window, and we will have an oval window. Okay, the oval window is going to be going towards your vestibular apparatus, which will be our semicircular canals and stuff. The round window will be going to the cochlea. Another name for the round window is cochlear window. Another name for the oval window is the vestibular window. Then also this will be drained by your pharyngotympanic tube, also known as the auditory tube. So, in the inner ear, there's going to be a bony labyrinth. It's going to be tortuous channels in the temporal bone with three parts. It'll have your vestibule, your semicircular canals, and your cochlea. Cochlea is going to be your organ of hearing. Semicircular canals will be for balance, and vestibule will also be for balance. There will be a series of membranous sacs within the bony labyrinth filled up with this stuff called endolymph. There will be three small bones in the tympanic cavity, and those are the malleus, the incus, and the stapes. So malleus is in hammer, incus is in anvil, and stapes is in stirrup. So the malleus looks like a hammer, the incus looks kind of like an upside down anvil, and this looks like a stirrup. Basically what happens is those guys, when sound waves come in and vibrate the tympanic membrane, these guys vibrate and they will amplify the sound to send it in to get processed. So next we're going to talk about this vestibule. It's going to be a central egg-shaped cavity in the bony labyrinth. It'll have two membranous sacs. There's the saccule and the utricle. Okay, saccule is going to go to the cochlear duct. Utricle is going to go to the semicircular canal, or it's going to be continuous with the semicircular canal, and this one will be the cochlear duct. Within these sacs, they're going to have maculae. Those are going to help out with equilibrium. Okay, so here's your utricle, here's your saccule. Utricle within the vestibule and saccule. This one's going up against the semicircular canals. This one's going up against the cochlea. Semicircular canals, so they're like basically like D or C shaped rings. They're going to be going in three different directions. Cochlea, it looks like a conch almost. And these two together will make up the macula. With the semicircular canals, there's going to be three of them. There will be one going anteriorly, laterally, and posterior. Each will define 2/3 of a circle, so like I said, it could be like C or D shape, however you want to describe it. There'll be membranous semicircular ducts lined that are going to be lined, and they're going to communicate with the utricle. Alright, inside them they're going to have ampullae that are going to have equilibrium receptors called crista ampullaris, and those receptors are going to respond to angular or rotational type of movements. With the cochlea, that is your organ of hearing, is going to be shaped like a conch, and it'll extend from the vestibule. It's going to contain the cochlear duct, which will have your spiral organ of Corti. That is going to be where the hearing happens, and it'll end at the cochlear apex. The cavity of the cochlea is going to have three parts: the vestibuli, the medii, and the tympani. Scala vestibuli, scala medii, and scala tympani. The vestibuli will go up to the oval window, and it'll have perilymph. The medii is going to be in the middle, and that's going to have endolymph, and the tympani is going to go up to the round window, and they'll have perilymph. So let's look at it here. Here's your vestibuli with the perilymph, the medii, this has endolymph, and the tympani, which will have perilymph. The roof of the cochlear duct is going to be a vestibular membrane, and the floor of the cochlear duct is going to be made up of bony spiral lamina and a basilar membrane, which is going to be the support, or like the basement, for the organ of Corti. The cochlear nerve is going to run from the organ of Corti straight to the brain, so this is going to be the main input place for that type of, for your hearing. So when you hear, first off, sound waves come in through the external auditory meatus. They vibrate the tympanic membrane. The vibration of the tympanic membrane will vibrate the auditory ossicles. The auditory ossicles will amplify the sound, and we'll have pressure changes within here. Okay, pressure waves created by the stapes pushing on the oval window are going to move through the fluid of the scala vestibuli. Okay, sound waves with frequencies below the helicotherma are not going to excite hair cells. That means you don't get any hearing. Alright, sound waves inside our range of hearing, they're going to vibrate the basilar membrane, they're going to deflect the hair cells on the inner on the inner ear, and you will perceive sound. With the auditory process, you're going to get impulses from specific hair cells, and those will be interpreted as pitches. So you can have your low pitches, high pitches, or you get the people who can, I can't tell you what note somebody's playing, but you can see exactly what note or what pitch something is going at. With loudness, that is going to be detected by the number of action potentials that will result when the hairs are deflected, and how large of a deflection those hairs are undergoing. When you want to localize sound, that is going to be dependent on the intensity and the timing of sound waves as they're hitting one ear to the next. With homeostatic imbalances of hearing, you can have different types. You can have conduction deafness, or you can have sensorineural deafness. Conduction deafness can be something like you have earwax and it's not letting the tympanic membrane vibrate. Or you could have sensorineural deafness where the actual nerve cells are dead or have some sort of damage to them. You can have tinnitus. Tinnitus would typically occur if there's some sort of nerve damage where the nerve is hurt but it's still working, or it could be from inflammation. There's something called Meniere syndrome. Meniere syndrome is going to be something that's going to affect the cochlea and the semicircular canals. So this is going to give you like huge dizziness. You just feel like you're spinning in a circle, and it's going to cause great nausea and vomiting. Okay, so our equilibrium and orientation. We'll have our vestibular receptors, which will be for static equilibrium, and we'll have our semicircular canal receptors, which will be with dynamic equilibrium. So dynamic equilibrium will be in our semicircular canals. Static equilibrium will be right here in the saccule. With the maculae, you're going to have sensory receptors for static equilibrium. You'll have one in each saccule wall and one in each utricle wall. They'll monitor the position of the head in space necessary for control, and they're necessary for control of posture. They will respond to linear acceleration forces but not rotational. So left to right, front to back. They'll contain supporting cells and hair cells, and the maculae in the utricle will respond to horizontal movements and tilting, and the maculae in the saccule will respond to vertical type movements. How do we activate these? By bending the hair cells. How do we bend the hair cells? When you move the head, there's fluid in there, and it'll push the hair cells away. We have a nice picture of it here. So depolarization occurs. So basically, you're in neutral, you're in midline. Nerve impulses are going to get generated in the vestibular fiber. If the head bends towards what's called the kinocilium, the hair cells will depolarize, and that'll generate more frequent action potentials. You put your head the other way, the hairs bend away from the kinocilium, the hair cells hyperpolarize, and they inhibit the nerve from firing. We have our crista ampullaris. There's going to be one in the ampulla of each semicircular canal, and these are going to be the major stimuli for rotation or rotary type movements. These guys are going to respond to bending of the hair cells. So if you depolarize it, you get rapid impulses to the brain. If you hyperpolarize it, you get less impulses to the brain. So here's somebody standing still. This is a standing still. As you start to move, rotation is going to start moving the endolymph inside the semicircular canals, and what will happen is the endolymph will bend the cupula and it'll excite the nerve hairs. Now check this out. If you're rotating super fast, what happens? This fluid gets moving and you stop and it keeps going. So as rotational movement slows, endolymph keeps moving in the direction of rotation, bending of the cupula in the opposite direction from acceleration and inhibiting the hair cells. So as you stop moving, it keeps going, but it slows down. With developmental aspects, okay, so we're going to have all special senses. You're born with them all at birth. Everything's present when you're born. Chemical senses, you could get a few problems that will occur in the fourth decade when sense begins to decline. So as you start getting older, you start losing your sense of taste a little bit. Not always, but it typically will happen. When you're first born, your vision is not fully functional. So newborns, that is why because those fibers aren't completely myelinated yet. So newborns can just see kind of blurry images. Babies are what are called hyperopic, so they'll only see gray tones, and their eye movements are not going to be coordinated yet, once again because those fibers are not yet myelinated. Depth perception and color vision is going to be very well developed by the age of 6, and kids are considered farsighted until they're about 6 years old. And with age, your lens can lose its clarity, those dilator muscles aren't going to be as efficient, and your visual acuity starts to decrease drastically, typically by the age of 70. That's it.